after the early morning's experimental session, I'm going to switch gears and talk about our computational work. So for the past about 10, 11 years, my group has been working on membrane protein structure prediction and computer-aided drug design. So what I'm going to talk about today is a computational tool we recently developed for drug discovery. So as we know, drug discovery is a very costly process. Shown here is some of the related data. So on average, it takes about 10 years and $1 billion to have a drug from starting basic research all the way to the market. So as we know, chemoinformatics and bioinformatics, they can facilitate drug discovery, particularly on the preclinical stage, to help reduce the time, reduce the cost. So chemoinformatics and bioinformatics, they can facilitate drug discovery in several different ways, as shown on here. The first is to help identify the drug target. And the second one is target modeling. So we know lots of protein structure is very challenging to solve experimentally using either X-ray crystallographer or NMR spectroscopy. But using computational tools, we can do this in a relatively quick time. And very importantly also, this chemoinformatics can help with screening the lead compound by different computer-aided tools such as docking and ship-based or physical-chemical property-based comparison. And further, once you have the lead compound, you can further develop it using computer-aided tool into a novel compound. So using chemoinformatics and bioinformatics to help with drug discovery, there's a remaining challenge called data integration. To help illustrate this point, I have two figures here. So on the left shows the total number of unique databases, bioinformatics tools collected in bioinformatics link directory. So you see here by 2013 or 2012, there's about almost 1,800 different tools and databases. And shown on the right illustrates the growth of GenBank in bases. You can see many of those tools, just like GenBank, they grow in an exponential speed or rate. So it's very challenging. So like I said, we work as a bioinformatist, but even for people in my group, it's very challenging to follow all those databases. So for regular biologists, they are not trained as a bioinformatist, but they want to do some basic drug discovery. There are almost no way for them to follow all this development. So it's becoming, I think, gradually important to develop further integration tools and other visualization tools to help people, help regular biochemists, biologists to get the up-to-date information about all those databases. That is a goal we are trying to do here. So for data integration in bioinformatics, if you look at the literature, there are several different approaches people have developed. The first one is called service-oriented. The idea is to do data integration through computer-to-computer communication. So you use web APIs, you do up-to-date data retrieval from diverse databases, and then you present them. The second main approach is called data warehousing. So by the name, it means they collect the data from different databases, then put them in a single database. So it's really like you go to shopping at Walmart. The Walmart claims they are one-stop shop. Means you go there, you can pretty much find everything you need. So data warehousing is the similar idea. They collect all the data, they put it in a single database. The challenge or the shortcoming of this approach is it's very hard for them to do this every day. So often you have a lagging effect. They may update their database, say, every week or every month. So for our purpose, we want to develop a tool like the biologists they can use, like they come to the office in the morning, they can open this tool, quickly get the idea what's new development to their protein of interest. So we decided we are going to take the first approach to the service-oriented web approach. So what I'm going to do for next part is briefly talk about the development of this tool we call PyMine. When we develop this tool, my personal philosophy or my guiding principle is we are not interested in developing something from scratch because there's lots of numerous people that are working in bioinformatics field. There's lots of beautiful tools already available. So what we want to do is develop something we call it value added. We are going to use the available tool widely used, then add something new to it. So as our graphic display, the tool we choose is called PyMol. I think from my discussion with some of you are aware of PyMol. It's a very easy to use molecular visualization system. It's also open source, so you can do lots of plugins, add different things to your PyMol. It's a leading software package, and we compared several, there's others like VMD or UCSF, they have this sorry. So sorry about this. When I'm in the US, I actually don't use cell phone. So I came here, I found everybody use cell phone. I decided to borrow one, but I really don't know how to turn it off. Sorry for the interruption. So anyway, back to PyMol. I think it's a leading software package like Camiro. Some of you may be familiar with the one developed by UCSF. But we after looking several of them, we feel PyMol seems very easy to use, probably widely adopted. So, like I said, there are thousands of different databases. There's no way in the initial stage we can integrate all of them. So our second guiding principle is we are going to focus on those comprehensive, high quality. High quality means they are maintained normally by government agencies, like by NCBI and US or European Molecular Biology Laboratory by European unions. Because those databases, they are updated more oftenly and they maintained regularly. So lots of individual academic labs, they don't, today they get funding, they develop a database. Tomorrow they lose their funding, then their database become obsolete. So that's our goal. We want to focus on those high quality, highly regularly maintained, and also most relevant to drug discovery. We don't want to aim for whole biology database too. That goal is too ambitious, we think. So the first database we focus on is called UniPro, which some of you may have heard of that. It's universal of protein. So it's a collection of protein sequence and function annotation along with links to different databases. The second tool we focus on is PDB. It's a macromolecule structure database, so protein data bank. The third one is CH, Chemistry of European Molecular Biology Laboratory. So it's collection of all the small ligand developed or biochemically tested targeting different proteins. So another one we choose is IBS, is a protein functional site annotation database. So the entries there is either experimental determined functional site or predicted. The phase one called HarmSaver is a database under NCBI, collection of human polymorphism and mutation data. The last one is a pathway. I guess we all heard of it, already developed at Japan. So we take these 6 databases, and the next one shows how we integrate them together. For drug discovery, we know if you know the structure, that we can do structure-based drug design. Or if you know the sequence, you can try to predict the structure. So the link underlying those 6 databases is either the PDB ID of the wild-type protein sequence, or you can use your own UniPro ID plus your own structure. Then we use REST API, which is a representational state transfer. It's a representational state transfer technology, basically web-to-web communication. So underlying this, we can link all these 6 databases, and then we can load the related information to PyMol for visualization. So eventually, our goal is to automate further drug discovery steps. So PyMine currently is writing in Python. It's becoming gradually and very popular bioinformatics language. We already tested it in Linux, in Mac, and Windows platforms. So here shows you the interface and the output. There are 3 parts when you open this plugin. On the left is we call the data panel. So the top is you can enter your PDB ID, or like I said, your UniPro ID of the protein, along with the structure in PDB format. Then you can look at the protein, basically the PDB file in text format. Look at the ligand. If there are any known ligand, you can get the smell format of the ligand. You can get the UniPro entry. So look at all the collections in the UniPro dataset. And like I said, PDB. Also, you can get the pathway from CAG. Very interestingly, we have a similar ligand function. So you can, if you know one ligand binding to your protein of interest, you can plug it in, click on find a similar ligand, then it will report all the non-similar ligand. So this is the graphic part in the middle. Basically, once you input your PDB ID, your structure, protein structure, will be automatically loaded. And also, if the ligand is binding to the protein, it will be there as well. Now, along with that, you have mutation data from like a SNP information, and also binding site. So you can basically toggle among these different buttons. Then you can turn on or turn off binding site mutation data, like a single amino acid variations, the ligand, see how they interact with the protein, or understand how those single mutations affect the protein function. So finally, it will automatically output some files, like a list of FDA-approved drugs and a ligand in standard file format. Also, you can output the pathway map if you are interested in look at the pathways. So those files are output, like I said, in standard format. You can use them directly for docking or other drug discovery effort. So for last couple of minutes, I'm going to briefly talk about a single case study. We used PyMine to done. Okay, so this case study focusing on an important class of enzyme called malic acid. It's a major enzyme in the production of NADPH. NADPH, we know, is a major reducing power for biosynthesis and for antioxidant defense. So it's also closely related to TCA cycle. So it's a central metabolic hub. In mammalian cells, there are two types of malic enzymes have been identified. ME1, ME2, that's what we call them. My collaborator, Dr. Xiaolu Yang's lab at the University of Pennsylvania, they reported a very interesting regulatory loop between P53 and ME1 and ME2 malic enzymes. What they found is P53 inhibit expression of these two enzymes, therefore suppress metabolism in tumor cells. That's one function of P53. But unexpectedly, what they found is also the downregulation of these two enzymes will boost the activity of P53. Therefore, it also determines the outcome of the cells. So that's a very interesting hypothesis. If we develop inhibitors targeting this enzyme, therefore regulate their function, then maybe we will help activate P53 in tumor cells to achieve the purpose of tumor suppression. So that's their hypothesis. And earlier last year, we get a pilot grant to explore this possibility. So when I look at this, when we started, I obviously have no knowledge about ME1 or ME2. I actually just learned from him. So when I use PyMine to look at this enzyme, I quickly get those information. I found out there's a crystal structure for ME1. I found the catalytic site by looking at the NADP H binding site. Also, I found the known inhibitors. So it literally took me like 1 day to get all this information, basically all the knowledge people have accumulated in different databases. Then I can easily come up with a game plan. Since there are known inhibitors, I can do ligand-based drug design using the inhibitor, using a software called SHIP signature developed by my colleague. So look at the similar ligand to those inhibitors. Then I can take all the similar ligand, do the docking study using this structure based on this catalytic site, okay, to get some idea whether those ligands will bind into the enzyme tightly or not. So finally, once we identify some top binding the ligand, we can do assay testing. So this is ongoing work, and what I've shown next slide is some of the top ligand we identified based on the docking study. First, based on the ligand similarity search, then do docking. So here you can basically from the zinc database, you can purchase those and do the test. So you see all of them are commercially available. We get a fairly good docking score. It's not that ideal, but that's the best that we found, and we are currently testing them. Hopefully we can find one lead compound for further design. So to summary, we develop innovative automated tools to help us integrate and visualize different chemical biology data for drug discovery. Currently, it integrates 6 high-quality databases. And also, you can output different files in standard format for the drug discovery project. So for future development, we are going to add more databases. Also, we are interested in doing some data mining work, actually. Just give a presentation at another university. They asked me this question. When you have all those databases, you collect all those informations, can you do some mining study to get the most relevant information? And last one is we want to integrate our current plugin with other tools so you can automate the entire drug discovery process. Right now, we only collect the information. Now, next step, we want to shape-based search than docking. So you can basically, from beginning, load your PDB ID. In the end, you get a list of ligands to test. We think that will be useful for biologists. So finally, I thank lots of people who worked in my lab, not necessarily on this project. I have several postdocs. Dr. Andrew Ham actually worked on part of the drug discovery. This tool, PyMine, was written primarily by this graduate student. Taiji Shui also worked on part of the drug discovery. And we have lots of collaborators over the years. Our work has been supported by different funding agencies. So I think that's my last slide, and thank you very much for sitting here, for your patience.
We have time to take a question.
Thank you very much, and thanks for the EU bioinformatics guys to help us to find our solution. I just want to ask quick 3 questions. As you are bioinformatics, so you need some chemical experimental result to develop this software, because as we know, the drug discovery is sometimes is pure chemistry.
Right, yes. So that's why we included the CHEMBL. That's the small molecule ligand database.
According to your knowledge, you think in the future it's possible to reduce like a chemical manipulation in the lab? Instead, you can do some predict result, because bioinformatics we know is very powerful tools in the science now.
I'm not sure what you mean prediction. If you mean prediction the binding affinity of the small molecules, is that what you mean, prediction?
Like prediction, for example, when you use bioinformatics, sometimes you can design some like a drug structure. I don't know, well, this field. Maybe, for example, for new disease, you can predict what you will use with some drugs without doing chemical manipulation in the lab.
Oh. Okay, so that's a very good question and an ambitious goal. I'm sure pharmaceutical industry will love that. But for drug discovery, obviously, if you identify disease, the next step you have to understand is physiology, its mechanism. Then you will identify a target. After that, you can use computer-aided tools to identify ligand binding to your target. So through that process, you have to do each step. I think using bioinformatics, obviously we can help with target identification. We can help with ligand selection. But I'm not sure we can really, just given a disease, a new type of disease, we can know which drug to use.
The last one. So you think, according to your knowledge, in the future, you can have some, like a chance for another researcher to do some update as we do in NCBI that can contribute for, share the main knowledge, something like that?
Oh yeah, for sharing, yes, we are actually submitted the revised manuscript for this one, and there we already put all the PyMine software manuals on a publicly available, I think, a website. So every one of you can just go there, download them.
Okay, thank you very much. I'm sure you can give a good contribution for the science.
One more question.
Yeah, Professor, I just wanted to ask, this shape signature, is it a part of PyMine or is it a separate program?
Shape signature and GLIDE, they are both individual standalone software.
Standalone, okay.
Yes.
And
so our goal is hopefully we can integrate them, but there are some issues with Python.
okay, and it does your screening of the database. You can use certain inhibitors which are already known, and you can use them to search the database for shape similarity.
Yes.
That is what it does.
Yes, that's
so it can use to screen Zinc database also.
That's the ship signature. That's what
Safe
that
signature,
design.
safe signature.
Yes.
Yeah. You can use safe signature
Yes,
for
yeah.
database searching.
It's very easy to use actually. There's a web server for that.
So like how long it takes for you to make
Oh, it's I think it's several hours.
because Zinc database is like to the tune of 15 million molecules.
Right. Yes. Yes. So this ship signature is my colleague's work, but what it does is convert a three-dimensional shape of the ligand into two-dimensional histograph. So the search is very fast.
They're using a histograph,
Yeah.
basically a hash print or a
Yes.
fingerprint.
So you are comparing 2D instead of
So
3D.
that was more quicker.
Yeah, that's very quick.
Thanks.
Yeah.
Last question.
On your first slide, can you show your first slide?
Sure.
The second one, that. The one where you should have Clara, number 4, I guess. Number 4. Number 4.
This one?
Okay.
Yeah.
How many of these databases are there right now?
How many? Okay, so if you look at here you are talking about this, right?
This is the tools, number of tools, right?
The number of tools and the databases.
I see.
So your question is how many of them are databases?
Who are active right now?
Active?
Yes.
Oh, that's another good question. So like I said in the beginning, even as a bioinformatist, there's no way I can follow all of them. So I don't know, but I take this data, all the data in this graph are taken from bioinformatics link directory, so which is a journal of bioinformatics. I think they have a collection, they have a directory. So that's the number of I take from there. But I didn't go through all of them to find out which one is active or not. So the recent Journal of Bioinformatics, they have every July, I think, or June, they have an issue of databases. So there, for this year, they have collection of 179. But I think many of them, they probably still exist. It's just that they don't update. Therefore, that volume won't collect them. Yeah. Thank you.